Swedish University of Agricultural Sciences in Umeå, and I will talk about classifying sex of ungulate species from camera trap images. So a bit about the background and the project. So at the moment I'm involved in a project where we are working on developing an online platform called Wildbild, so collecting organized camera trap images that are collected by hunters on the national level. And we want to use this as a digital monitoring tool for Swedish game species, like getting information on distribution, density, reproduction, survival, to ensure stable populations. But we encountered or experienced that there was some really dry spring and summer in the last years, also in Sweden, and this resulted in really low reproduction of those ungulate species. And one important background information that the hunting quota in autumn-winter in year T is based on the harvest and observations from the hunting season and autumn-winter from year T-1. So this means also that this whole reproductive season is not considered at all. And this can result in overharvesting when, for instance, in specific years, the reproduction is very low due to drought. So one potential solution would be to have cameras deployed in the field that give us images, and we can get a really quick classification of the age and the sex of those ungulates during late summer. And then we can take this into account, estimate some female juvenile ratios, so some reproduction estimates, and incorporate this in the estimates for quota for the next hunting season. And this would result in like a much shorter feedback loop between the monitoring and the management as compared to what we have now. And this can also result then in more appropriate management by including this reproductive output into these quotas. So my goal was, or is still, to build a sex and age model that classifies juveniles and adults and males and females from camera trap images across 4 ungulate species. In this presentation here, I only focus on the sex model. And our general approach is to have, we have first worked with mega detectors. So mega detector is an object detector. He puts like bounding boxes around objects and classifies or distinguishes animals, vehicles, humans, and empty images. Next, for only the images with animals, we use a species classifier, distinguishing or classifying different species. And then on top of that, for specific species, we want to have a sex and age classifier. So we use camera trap images that were collected across 5 different projects here in Sweden, across 6 different years, and more than 400 deployments, so different locations that you can see there on the dots on the map. We have in total 106,000 labeled images across 4 ungulate species: roe deer, red deer, fallow deer, and elk. And of those, we have 60,000 where we know the sex. And finally, we have 30,000 observations where we also have bounding box. So this is basically what we can use, what we have available at the moment. So I created a prototype, like a subset of these images, of these 30,000, which are like about 8,000 images, and we used a stratified sampling to make sure that it's representative for the whole data set. So we considered with that like having different deployments, like and day-night images, different month. Also the bounding box that were created by human or by mega detector. If we use mega detector bounding boxes, we only included images with observations with a score of larger than 0.9. So we split up the data set and train 70%, well, and test 15%. And he accounted for having sequences of images in one of the 3 splits. So sequence of images when the animal is walking in front of the camera, it not take only one image, but a couple of images, and the whole event is one sequence. And we also split by deployment, so make sure that we have whole deployments only in train, oval, or test. It's because we want, at the end, generalize our model for other locations in Sweden. And this is what Sarah was talking about, like the out-of-distribution issue. So just to make sure that our model is well performing also new data. We use supervised learning and the ResNet 18 architecture for binary classification. So we crop the images based on the bounding boxes, and we use as training hyperparameters what you see here, and I kept them across those experiments that I showed today constant. So to start with some results of the base model. So what you see here are different metrics. Here the mean average precision is 0.57, 0.66 for the accuracy, recall 0.45, 45, sorry, and can't read it. It's like 0.59 for F1 score. But when we investigated then the images, inspected the images, we saw that because we cropped them based on bounding boxes, we have really different aspect ratios, as you can see here. So we included some padding to make sure that, around images that were cropped that don't have the size that ResNet needs, we have some black space. This does not really improve our model a lot. So you can see here, it's the second bar. And then we also did some augmentations. I will go quickly through it because I don't need to do like too much detail. But what you basically see, we have done different augmentations, flipping, color jetting, random perspective, blurring, random grayscale, and the most interesting, so what I considered our best model would be this one, where we include the first four augmentations, which resulted in an accuracy of 0.72. And I focused more on accuracy because we are interested that the model is predicting the right thing, like for males and females, and not only for males would be like the precision in this case. So this is the result then across also different runs. So we have here like 5 runs that I did for this model, and we see that the model seems to be in accuracy quite robust, like somewhere around 0.7. Then to show you some confusion matrix of the best model, we see here that 76% of the females were predicted correctly and only 61% of the males. So it seems that it has some, model has more difficulties to predicting the males correctly than the females. And this is basically what you also see here in this histogram. So we have here the female prediction score. So here the females that were correctly predicted, and here the females that were wrongly predicted, and this is much less observations here than here for the male prediction. So if we look at, we also included the gradcam function to see what the model is actually looking at, and what you see here is the original image with the rhodea male, and overlaying it with this gradcam, you see here that the model is focusing on the antlers to predict that this is a male. This is a fallow-deer female, and here we see that the model is looking at, also the head, checking out if there are antlers, but also because there are no antlers, probably looking at if there's something at the penis. So these are some results, like showing the full image with the prediction. We have here, for instance, a rhodea male with a score of 1, a rhodea female with a score of 0.96, here a red deer male and a red deer female. So just to show you also some variation of how the images can look like. And an elk female and the rhodea male, which has much lower score. It's much more noisy, blurring image, much more difficulties to predict it. So if we have a look at the top 50 true negatives, which means like the females that are correctly predicted as females compared to those that are actually females that are wrongly predicted by the model, we don't see too many, I don't see really a pattern what's happening here. But what we see is that there are some of these, how I call it, like edge images, where we only see, for instance, here, it's only a leg of a deer. Here also we see only really some kind of body parts, and even if we would only get this image, we wouldn't be able to say what sex this is. If we as human label those, we always see the whole sequence of the animal passing by the camera, which makes it much easier than only getting these, one image of a sequence. So that can make it really hard, and we have to think about how we can maybe solve this. Another thing, what we could do to improve the model, thought about like, I did it now, the model across several species, maybe focusing on only rodeo, on only one species. And I compared those models and saw that the accuracy actually of the rodeo model is lower than for the whole ungulate model. But the precision was higher, so it seems that it was easier than to predict correctly the males. I also looked into maybe it's easier to only use a specific season, like only the reproductive season, what we're interested in. But here, I didn't really see any improvement at all in the metrics. So, yeah, at the moment I stick to this full across ungulate and a model for the whole year. So to summarize, the model at the moment is like performing like within 70% accuracy, I mean average precision of 60% to classify correctly males and females across 4 ungulate species. The species-specific model for the roe deer had higher precision, but the accuracy was slightly lower. I have to check for other species how that will perform, and the seasonal model did not perform much better. But I also encountered that I have to check annotations. So what can I actually expect from my model? How well can it actually perform? Because I also found some annotation errors, of course. So we have here this guy, which was labeled as a female, and we have here 2 guys, which were also labeled as female. So there are definitely also some errors in the data set. So, yeah, then I always think about how I can incorporate or maybe exclude this issue about the edge images. Maybe trying out some different architecture could work. And of course, remember from the introduction, I want to say something about reproductive output. So we also need to have a well-working age model that I'm at the moment working on. So, and with this, I want to thank you all for your attention, and especially Sarah, who organized the CV for Ecology summer school that I participated in August. And basically, this what you saw just is the result of what I worked on during the school. So, as a completely new ecologist to computer vision. So, thank you very much for your attention.